Hi class, today we're going to be looking at passive transport in the cell membrane. And so we have 5 questions here you need to be able to answer. The first is define concentration gradient. Number 2, what is diffusion and how is osmosis similar and different than diffusion? Number 3, explain equilibrium and how it relates to movement of particles across the cell membrane. Number 4, define and explain hypertonic, hypotonic, and isotonic as it relates to the cell. Provide one real-life example. And number 5, what is the difference between facilitated diffusion and diffusion through ion channels? Okay, so we're going to be looking at passive transport in this set of notes. We already talked about homeostasis and how homeostasis maintains an stable internal environment. And passive transport is going to allow that to happen. So passive transport is movement of materials across a cell membrane without using energy. Now, as a reminder, the energy molecule is ATP. And that should be a reminder from last chapter. So when you see the word ATP or the initials ATP, that just means energy. There are 4 types of passive transport: diffusion, osmosis, facilitated diffusion, and ion channels. Now, diffusion is the main topic here. Osmosis is just going to deal with water. Facilitated diffusion is just something helping things across the membrane. And diffusion through ion channels is similar. It's just specifically talking about ions that are passing through the membrane. So if you want to make a note next to any of these, you could. Osmosis is just going to deal with water. And then facilitated diffusion is going to be more like this here, where something is helping the particle to get across the membrane. And ion channel is going to be very similar to facilitated diffusion. So diffusion itself is movement of molecules from an area of high concentration to an area of low concentration. So from high to low. And this just happens naturally. No ATP is needed, no energy is needed. This is the cell membrane. And these are particles that have kinetic energy. So they're just moving along. They travel at whatever pace they happen to be going, solid, liquid, or gas. And they will just cross the membrane on their own without needing help. They just will pass right over. Some of these will be going this way, and they just pass through just because they fit. They're small enough or they are nonpolar. So the types of molecules that fit is small and nonpolar. Now they could be either or. They don't have to be both. So they could be small but polar, but as long as they're small, they'll fit through. It's like they're sneaky. They kind of fit through the membrane. Okay, so here's an example. We have high concentration to low concentration. The solute is what is being dissolved in the water or fluid, whatever it might be. And the cell membrane is that white line there. And all of the movement happens because of kinetic energy. So again, the solute moves down the concentration gradient from high to low. The concentration gradient is just a difference in concentration across the cell membrane. So one side is considered the outside of the cell, one side is the inside of the cell, and you're looking at how much solute or how much stuff is on the outside or the inside. Generally, through diffusion, things go from high concentration to low concentration because no energy is needed. It would be like falling down the stairs. You don't really need energy to fall, but you need energy to go up the stairs. You have to work a little harder to go up the stairs. Falling down, you don't use any energy. Okay, equilibrium is the goal of the cell. So this is the goal of the cell during diffusion. And the goal is that they will have equal concentrations on both sides of the cell membrane. equal on each side of the cell membrane. Once it has equilibrium, that is the end of diffusion. So at that point, the end of diffusion, there's no net movement. It's not like they're rushing over to that side of the cell membrane, but there is still motion of particles. And this is a key thing. There is still motion of particles. They're just still random, and they're not all rushing from one side to the other. Once they're in equilibrium, there's still particles going across the membrane, but they're just doing it randomly. So this is just an example of how that happens. Dye is dropped in. You can see the particles will all have motion. They're a liquid, so they slide past each other. Eventually, we get to an even distribution of the ink. So, a couple things here. Diffusion across a membrane will depend on size, shape, and chemistry. Things that are easy to diffuse are small and nonpolar. So to show you kind of what's happening here, we'll use this website. Okay, so after that little animation, you can see that particles are in motion and they spread out to fill their container or fill their space based on the kinetic theory of matter. Now, osmosis is just diffusion, but talking specifically about water. And the process is still down the gradient because it is still diffusion. So we're still looking at the term diffusion here, and it's still going down the gradient. But there's going to be a couple new terms here, hypotonic and hypertonic. Okay, so if we look then at those terms, hypotonic, hypertonic, and isotonic. Hypotonic means that the solute concentration is higher inside the cell. So water is higher outside the cell. And what can happen is that water will rush into the cell trying to even out that concentration. The cell will swell up, and in plants this will be called turgor pressure. It makes the plant stand up tall. It's kind of nice for a plant to have that. But in animal cells, you can see here that they could burst. So this is a red blood cell that had too much water rush in and the cell burst. Cytolysis is called cell death, and that's what can happen here. Hypertonic is when the solute concentration is higher outside the cell. So water concentration is higher inside the cell. And what will happen is water will rush out of the cell. In plants, this is a loss of turgor pressure, and the plant will wither. It can cause what's called plasmolysis, where the cell shrinks. And you can see here, this is a red blood cell that has shrunk because the water has left the cell. In an isotonic solution, solute and water concentrations are equal on both sides of the membrane. So water travels back and forth in equal amounts. It's called equilibrium. Water's still traveling, but it's in equal amounts across the membrane. We will be doing some lab information with this, dealing with potatoes and salt water versus regular water, and we'll be able to see this in action. Here's the example as well. Hypotonic, water rushes in. For a plant, that's great, makes the plant stand up tall. For animal cells, it can burst the cell. Isotonic, water's going in and out equally, and it's pretty good. And then in a hypertonic environment, water will rush out. It'll shrivel up an animal cell. It'll wither up a plant. To show you that in red blood cells, you can see the examples here. Isotonic, hypotonic, and hypertonic. You have this in one of your worksheets. You can fill these in if you have that with you to show you what it looks like for each of the types of plant cells and animal cells. We will continue with facilitated diffusion in the next recording.